Hi there, I'm Patrick Degenar and this is bioelectronics. So in this lecture I want to expand further on optogenetics and really talk about both the application space but also the key caveats to the optogenetic technique. Nothing is for free, unfortunately, in life. So first of all, let's talk about the application space, and this is a concept image that we could have some kind of future neural interface where optic fibers come down and connect into our brains and allow us to upload and download memories to the internet. Some of that's perhaps a little bit fanciful, but certainly being able to control the nervous system or parts of the nervous system will allow us to do things that we could not do before. Certainly in the beginning of the optogenetics field, one of the real beauties of the technique was that you could take neurons in culture and you could shine a light and say, wow, right, I want this signal for you guys, and I want you guys to be quiet now and don't have a particular signal. And you can see that in this particular data form here, which was actually taken in Newcastle. This is these dots here represent the background activity. From top to bottom, that this is the process run again at the same reference point, and the light shining at the same time from this reference, tended to get activity in each case. So this is beautiful. You can control the cell at will. And one of the other nice things about the optogenetics technique is that you can genetically sensitize specific circuits. So it's not just putting an electrode and stimulating everything, but you can specifically say, right, I want pyramidal neurons, or I want that particular subcircuit. So you're combining the power of genetic technology with interesting optoelectronic technology. So it's very powerful. But there are also caveats, which I will go into over the course of, over this lecture. So first of all, as I've said before, the beauty of the technique is, look, this was a very early paper. This was by Zhang et al., where they fired in the blue light, and for every blue light they could get a channel rhodopsin mediated depolarization action potential, and then they also had what was called halorhodopsin in place. So this was kind of an inhibitory agent, and they could prevent activity even when they were shining the blue light. So it's a very powerful technique. But let me come up with the first caveat, and that is light requirement. So the first caveat, let's compare the different types of optogenetic agent. So the first optogenetic agent at the top is the G protein amplifier type approach. The second is to use the channel rhodopsin approach, and the third is to use the halorhodopsin approach. So this is where it's a light activated pump. Now what these are graphs. So this is for the GPCR approach. This is for the channel rhodopsin approach. This is for the halorhodopsin approach. Now, unfortunately, people in the community use different, milliwatts per millimeter squared. Sometimes photons per centimeter per second. So let me translate that. So the melanopsins typically take about 0.4 microwatts per square millimeter. Channel rhodopsin takes 0.7 milliwatts per square centimeter, and halorhodopsin takes requires about 10 milliwatts per square millimeter to achieve what it needs to achieve. So clearly these are orders of magnitude different. In particular, melanopsin is about 1,000 times more sensitive than channel rhodopsin. So okay, this is great. This is fine. We can use melanopsin. But there are a few additional subtleties to that. Let's go back first of all to this light requirement. These milliwatts per square millimeter, etc. What does that mean in terms of actual values that you may understand. Well, the melanopsin sensitivity is about here, which, you know, if you convert the milliwatts per square millimeter into luminance, lux, it's about at the level of a kind of a dull, actually that's a pretty bright family room. Most you know, offices are around about 3 lux or so. But if you then look at channel rhodopsin, you know, channel rhodopsin's 1, which is way higher than an ultra bright day. You know, it would quite literally be like, you know, sitting in the Sahara Desert in midday bright sunshine. Halorhodopsin's even brighter. It's 10 times brighter than, ooh, you know, I mean, this is super bright as a requirement. So, you know, the primary forms of optogenetics are channel rhodopsin and halorhodopsin, for reasons I'll explain in a bit. But these require very, very high intensities of light. So that's a key caveat. But you may sort of ask, well, hang on a second. We see starlight as human beings, and we see starlight at effectively video rate. You know, what's going on here? And let's look at a human rod cell. So we have 2 types of photoreceptors. We've got cones, which are the kind of daylight cells, which allow us to see in daylight, but allow us to see in color. And we've got rod cells, which are night vision sensors in our eye. But let's look at the rods. Now, light will come in, well, sideways, if you like, or into our eyes. But let's imagine the light is coming up towards the rod. Now, all of those rhodopsins sit in these special regions, in special kind of little bubbles, if you like, which are the absorption layers. So, unlike a normal cell, the human rod has about 1,000 absorption layers. If instead you take a normal neuron cell, which we can treat like a spherical, you know, a spherical object, and you stick on an artificial opsin onto its surface, well, it effectively has 2 surfaces. It has the bottom surface and the top surface so the light can go through. The second thing is that the only purpose for these human rod cells is to sense light. So you have a cell body here. This is the body. And, you know, that performs, that's got the mitochondria and all, and the nucleus and all the components that, you know, keep the cell alive. But then you've got these special absorption layers whose only purpose is to absorb light. So they're packed full of all of these rhodopsins and their G protein cascade components, and, you know, packed full of the things that are used to sense the light. In a neuron, the surface has got all sorts of different receptors and channels and so forth. So you cannot pack the surface to the same degree. So if you actually look at the density of these opsins on the surface compared to the density of the rhodopsins on a human rod cell, they're somewhat different. I mean, neuron cells were never designed to sense light. We've just artificially modified them in this case. The third thing is amplification. So the channel rhodopsin, if we use a standard channel rhodopsin, doesn't, you know, just, it's a channel. Light goes in, channel opens up, you know, light enters the cell. If you use a use a light activated pump, it's even worse. It's just one photon absorbed equals 1 ion passed through to the other side. Whereas we've mentioned before that these kind of GPCR, you know, metatropic ion channels, if you like, you know, one absorbed photon will deactivate. It's weird, it's the other way around in humans. Will deactivate, you know, potentially hundreds or even thousands of channels. So you get this massive amplification. And then finally, the cell size. You know, all of the inputs from these, from these outer areas, will influence. It'll all stream down. You got the pumps, and you got then this hyperpolarization effect. So it all streams down to a small cell diameter, which is about 2 or 3 microns. Whereas in the case of neurons, the diameter may be 5 or 10 microns. So when you compare the two, given these effects, the cells that are in our eyes that can sense starlight, 10 to the minus 9 milliwatts per square millimeter, if we take these considerations into account, we pretty much get the 1 milliwatt per square millimeter, for a channel rhodopsin encoded neuron. So it's not something fundamental. There are a few things that we could change, such as the conductance of the channel. If you remember that G term in the Hodgkin-Huxley, we could change that G. But apart from that, that's pretty much what we've got. It's a fundamental caveat. Now, you may say, well, okay, hang on, but you said at the start that there were these melanopsins. We could use those. Now, it turns out melanopsins are special. They're actually in our human system. They sit in the communication cells in our eyes and modulate our awake-sleep cycle. Now, and they basically, they've got some connection with some ancient ancestor or directly with an insect, because they're basically the same thing as that used in insectoid or invertebrate vision. But the problem with them is, again, we mentioned before that if you've got these kind of rods and cones with the special structures, once you put this into a large cell, what we find is this. So this is a paper from early on in the optogenetics field. And what the authors did was they shone light for 10 seconds, not milliseconds, and then you got an effect, and you got a very strong effect, and hey, it was very sensitive. You only need a little bit of light, and you got this effect. But there was a 10-second delay. Our vision normally operates in 50 milliseconds or faster, really. So we need neurons to respond in the milliseconds or tens of millisecond speed, whereas the GPCRs, when they're programmed in, take a lot longer. And why is that the case? Our eyes are pretty fast. Why aren't these fast? Well, if we look at these special structures inside the human rods or the human cones, these are packed full of all of the components. So if you think of this G protein cascade, the receptor has to talk to the G protein. The G protein has to talk to the PIP or the CGMP or whatever the components are that turn on or turn off the ion channels. It's got to have them all close by. If they're further away, the probability of finding these things gets worse. Now, in these human or these dedicated vision cells, everything is packed close together, and the area is quite small. If you go to a neuron cell, and you got this big area, 10 microns, it takes a long time to find the components, and therefore it's significantly smaller. So if you look at the difference, it's a 10,000 times difference, and it's basically largely due to the areal difference as well as the packing density. So what does this all mean? Well, one of the other things that we've been exploring within the field, I'm not a molecular biologist on this, I tend to make the electronics, is how to shift also the colors. Generally speaking, the violet range is bad. We want to operate in this visible range. And one of the reasons for this, if you look at the energy of a photon, which is related to the wavelength, if the wavelength is too short, the energies get too high. And, you know, once you get into this kind of 2.5, 2.9 electron volts, they'll start to break things. They'll start to cause photochemical damage. So one of the key, moves in the field is to also make these opsin variants more towards the red color. So if we want to summarize this, optogenetics is a fantastic technology. It allows you to genetically encode cells that were never meant to be optically activated, and you can suddenly control them, remote control via light. And because it's remote control via light, there's potential there for less contact, and there's no electrochemical exchange and all sorts of things. So it's a very, very good technology. Certainly for science, it's been transformative. But, we have to be aware of the limitations. So our options in optogenetics are to use melanopsins. Melanopsins are very sensitive. You don't need a lot of light, but they're very slow. So they're probably not good for things like vision, which need to be fast, but they could be used for applications where, you're running in hertz or subhertz, cycles that happen once a day or once every few hours or once every 10 minutes. Channel rhodopsins are the primary form of optogenetics, in that they're kind of a mixture in between the fast, the slow, and but having reasonable sensitivity. It is worth noting that the light requirement of these are quite significant. It requires a lot more energy than electrical stimulation of cells, but in return you get this extra power, and the responsivity of these cells can be quite good, down to the millisecond with channel rhodopsins. Halorhodopsins were very exciting early on because they gave that negative capability. You could have a channel rhodopsin which will excite and a halorhodopsin which will inhibit. But more recently, we've had the advent of channel rhodopsins which are also inhibitory. And as such, probably halorhodopsins will not be used very much in the future. They're very fast, but just simply they need far, far too much light to be activated. I will talk in the future lecture. So this is really about stimulation of neurons. And there's actually a way to also use these kinds of techniques to record from neurons. But I'll cover that in future lectures. So this is it for now. Thank you very much, and see you next time.